So welcome to the next part of the demo session, which will now cover RICS, or as it stands for Raster Image Correlation Spectroscopy. So I will explain the technique in a bit, but first take a look at the microscope that we have, which we use for the correlation spectroscopy tool. So this is the LSM 880 size microscope. We have, as you can see, it's an incubated box, so you can do your imaging at 37 degrees or your spectroscopy measurements at 37 degrees. And then, so this method is extremely sensitive to temperature. So if you want to measure it at 37 degrees or 25 degrees, you need to do some sort of reference spectroscopic measurement to sort of have your parameters ready, which I digress, which we'll get to in a little bit. So as you can see, I have already placed my sample inside, and today I'm going to be showing you two different kinds of sample. One is EGFP in cells. The second is my protein of interest, which is this one. So this one is a disruptor in schizophrenia 1. It's a major protein that's involved in mental illness. So I'm trying to understand so many things about this protein, and in today's demo session we will focus on how this protein diffuses. So that would be your actual experimental sample, and I have my disc 1 fused to EGFP, so that's why my control sample is EGFP in cells. So we have our laser lines coming from the back into the microscope, and we have our scan head right there, and we have our detectors right here. So this is everything that you need to do RICS. And of course, you need an expert, so you need to become one, and I'm going to try to explain to you the technique a little by little. Now I'm going to show you how the experiment is done. So for this, we are microscopists, so we definitely need the lights on. Okay, so what you see here, I've already made like a snapshot of what you can actually see through the objective, because I can't really show you. I'm just showing you this. So what is the first thing that comes to mind, or what you can see is you have cells which are transfected with different transfection efficiencies of EGFP, sorry. And for RICS, we tend to avoid such cells which are extremely bright or intense with transfections because of crowding. We cannot really estimate to a good amount or to a good extent as to what the diffusion looks like in such cells because of extreme molecular crowding. We like to choose cells like this, preferably, or maybe a little bit better than this. So I'm still not convinced that this is a very good cell. So what I'm going to do is I'm going to take a much broader picture. So for this, I'm choosing a tile scan of 5x5. And I just hit start experiment, and as you can see, cell starts to appear. So this is an overview of the 5x5 tile scan that I just made, and I think I really like this particular cell. This is a very dim cell, so I'll just move this box for you to see. So this one, and I'm going to focus on it, and I'm going to hit on continuous, because I think that is a very good cell for analyzing RICS. So it's this one. So you can clearly see that this is a GFP cell, because you don't really see the nucleus here, because the expression level of EGFP is almost very similar in the cytoplasm as much as it is in the nucleus. So what I'm going to do is I usually measure the RICS 4 microns above the surface of the membrane for a cytosolic protein, and this is a cytosolic protein. So I'm going to go focus on the membrane completely. So I would call this the membrane, and I'm going to go like 4 microns above the surface of the membrane. So, and this gets me here. And what I'm going to do right now is to put the pixel size to 50 microns, because that's my predefined parameters I use for my analysis as well. So I already know that I need to zoom in 10.5 times to reach 50 nanometer pixel size. I click on the region of interest, so I'm guessing the cytosol is somewhere here, looking at the structures. So I'm taking a little bit of the nucleus. I'm sorry. I see the nucleus is here. So I'm going to take a little bit of nucleus and a little bit of cytoplasm here and zoom in 10.5 times and hit continuous. And I have to do best fit. So how you know that this image can be correlated, or this cell can be correlated, is you see very closely. If you notice, you see horizontal lines moving, and you also see this line scanning. So what this line scanning means is that it means it's a raster scanning. So raster scanning basically moves from left side of the pixel to the right side, and then back to the next line to the left, and then goes again. So this kind of zigzag pattern of scanning is called the raster scanning, which is what is happening here. And now what you would now see me do is I will very quickly record this for 50 cycles and try to get the diffusion coefficient of this cell. So parameters adjusted, and now start. So another important point to mention here is that I use photon counting mode to do my analysis. So and while this is recording, I will quickly explain the setup here. So I have taken EGFP, so I'm measuring EGFP, so I'm exciting with a 488 laser. Yeah, and I've put my detection from 489 to 633, so the complete range of EGFP using, so I'm scanning for the laser with a 488 filter, and this, what you see here is the spectral detector here, so that's where we collect the photons. Okay, so moving on to the next sample, we have a cell that's transfected with mouse disc 1. So normally disc 1 transfections are characterized by the presence of these higher order oligomers that you can actually see as these blobs which are in motion. So, and we think that these are higher order oligomers, which we will exclude from the analysis of disc 1 diffusion, because they will definitely hinder in the process of analysis. So again, the same protocol applies. Find a cell that is good enough for doing RICS, and you go down all the way to the surface, to the membrane, and go 4 microns above the surface of the membrane, and then there you have it. Same rule applies. You set the pixel size to 50 nanometers, and you choose a little bit of the nuclear region and a lot of the cytosolic region, because disc 1 is a cytosolic protein. So, and you're majorly interested only in analyzing the diffusion in the cytosol. You include a little bit of the nucleus just to have a sense of the orientation of the cell. And then I again record like 50 cycles for this. You press continuous to make sure it all looks fine. Yeah, now you can see that these higher order oligomers become a lot more prominent. Now I am just going to record for the next 50 cycles in this. We now saw how easy it was to record the RICS data. So in this part of the demo, we will now look at how to analyze the data. And to analyze the data, we use this MATLAB-based software. It's a home-built software. It's called PALM. It's downloadable easily from Git. And there is a document called Read the Docs for PALM, and there's a step-by-step procedure as to how to download the data. Okay, so now we recorded one sample each of EGFP transfected in cells and one of
Disk 1 fused to EGFP transfected in cells. Let's now load the EGFP data, which is this. And when you load the data, it's also very simple to load. So the first thing that you can notice is how you have a display of the average frame count rate per frame. So you have frame in the x-axis and the average count rate in the y-axis. Now, what you look at here is how the cell looks like, and to get an overview, you go, oh, this is a summed image, and to get an idea per frame, you scroll through the frame, and you normally do this for all the cells to just make sure that your cell or your file is not corrupted. So this is the first step. What is to be done is you have to correlate every pixel across frames and get what you call as a correlation map. To do that, it's important to include only the regions which you want to include for correlation. So, what are the regions that you normally exclude for correlation? It's these regions which are extracellular regions, which is normally excluded from the analysis because it doesn't correlate to anything. Also, if you notice that there are aggregates in your cell, you normally exclude those regions as well from the analysis. So, this program remembers the last cell that you analyzed and it displays the same values for intensity thresholding, as we call. And because of that, you also see some nice pixels being excluded from the data. So the red region represents the pixels which are excluded from the analysis. Uh, Giving a reset just resets the whole data. I'm not going to explain too much into this analysis, but I'm just going to show you how to do an intensity threshold. I'm going to give 200 and to see if just the region that is extracellular is excluded, and it automatically gets excluded, which is nice. And you don't need anything else because you don't have an aggregate in this cell, or so I remember. Okay, so the next step would be to just press do Rix, and it will save. It will get saved. You normally open in the Rix tab to see how the correlation function looks like. I will get in a minute to interpret these results in a while. But in the meantime, I will also repeat the same thing for the mouse disk one. So I'm loading the file. So what you see here in this case is this is the extracellular region, or no, I think this is the nucleus. So this is the nucleus, beg your pardon. And what you also see here are these aggregates. And these aggregates can cause aberrations or it can cause inconsistencies in getting the correlation function right. So what you do is you also threshold these regions, and to threshold these regions, which are very high in intensity, you use the maximum channel. And now I see that on an average it's about 600. So this pixel is slightly more than the average intensity. So what I'm going to do is I'm going to give it 800 to see how it gets thresholded. I also see that in some cases, pixels like this also get thresholded, which is not entirely necessary. So you increase the intensity to exclude the low intense regions to exclude from the analysis. This seems to be okay. I'm going to give 1100, and to me, this seems to be the right way of thresholding it. Okay. Also, I noticed that the nucleus is not thresholded, so I'm going to increase a little bit on the minimum to see if that solves it, and it does. So the next step is to again go through the frames to sort of make sure that the file is not corrupted. In fact, this is the first thing you do, but I already did a quick test before I started recording this demo to see if the file was okay, so I think I got carried away with that. Okay, good. So I'm gonna click 0 here. All right, and now I'm gonna save this file as well. Okay, it says it's done. And you automatically notice that this correlation plot looks slightly different from what you saw for EGFP. To understand this plot, let's open what is called Mia Fit. So you fit the data, and I open my correlation file. So I did this today. So this is for EGFP. I'm going to load this, and I click on the active channel. So, we already saw before that the width of the laser beam was all optimized, and this is what you get from that: the radius and the width of the laser beam. The pixel size is 50 nanometers, which is constant and it's fixed. Um, and this is the waist size, which is 1.5. This is normally quite standard, and it's also optimized for each objective. The pixel dwell time is 8.19 microsecond, which I mentioned it to you, and this is the line time, which is 4.92 milliseconds, which is the total time of the scan divided by the number of frames, which is no, the number of lines, which is 256, and you get this. So it's all very simple now. You just start, fit the data, and what you see here is the diffusion coefficient of EGFP is 20 approximately micrometers square per second. So we already know from literature that the value of diffusion coefficient of EGFP is anywhere between 20 plus or minus 5 micrometers square per second in a diffusing when it's freely diffusing in cells. And this pretty much falls right in the range, which so you can really accept this value. What you also notice is how it shows you the number and brightness, which gives you a fair idea of the oligomerization state of the protein. And for EGFP, it's normally around 5 kilohertz, which seems to also be agreeing with the literature here. I'm gonna have this just like that, and I'm just gonna add a correlation file, which is the mouse disk one, and I'm gonna I'm gonna inactivate that, and I'm gonna activate this so that it shows you now here, and so that I can shuttle back and forth and show you how both of them look like. So you automatically notice that the correlation pattern is different. So to help you understand this, I'm going to first fit this data as well, and I'm going to let you think as to what it can mean. So look at the diffusion coefficient here. For EGFP, you get 20 micrometers square per second, but for mouse disk 1 that's fused to EGFP, you get 0.35 micrometers square per second. So, what you can immediately notice is that the diffusion coefficient is vastly reduced. Now, why could this be? Could it be because EGFP is a bigger molecule, and when it's fused to EGFP, that the size gets so big that it starts diffusing slowly? Maybe, but we already know that for making up to the size of EGFP fused to disk one, you need five EGFP repeats, and we actually repeated that. So we tested the value of the diffusion coefficient of that, and we got a value of about 6 micrometers square per second, which is still faster than EGFP fused to EGFP fused to mouse disk one. So what do you get from this? How do you interpret this? So look at the brightness. It's almost 1.5 times or 2 times higher than just the EGFP value. And like I said, brightness is an indication of oligomerization. So from this protein, from this that this protein is oligomerizing and it's diffusing as oligomers. and that's why the diffusion coefficient is also vastly reduced. And what do you interpret from the shape of the correlation? So here you have the x-axis, you have the y-axis here. So pixel lag in Z, pixel lag in Z. So you see that there is some sort of diffusion pattern along x and along y. While if I move suddenly switch to EGFP, you only notice that there is diffusion along x, but there is no defined shape along y, which is a clear sign of a fast diffusion over a slow diffusion. And RIX is a very cool technique to help you understand the nature of the diffusion with by just even of an unknown sample, and you can just do the right control measurements to really understand how fast or slow a molecule diffuses in a cell. And I think with that we conclude the demo session.